Okay, thank you. So yeah, today I'm excited to present work together with Heinrich Hoffmann, where we discovered TMEM41B to be important for both coronaviruses and flaviviruses. So first, I'd just like to start by addressing the question, why do CRISPR screens and what are we looking for? So to introduce this, I'll just go quickly through the coronavirus life cycle, just to highlight the point that viruses in general and coronaviruses as well rely on host factors throughout pretty much every stage in their life cycle. So this is from the very first steps of receptor binding. We know that SARS-CoV-2 engages ACE2 on the surface. They utilize host proteases to fuse and then endocytose. Once the RNA is delivered into the cell, the virus relies on host ribosomes and other proteins for translation. These proteins are then processed, and then they hijack cellular machinery in which they form replication organelles, which are lipid protected, to replicate and make additional RNA copies, and then mature and egress through the cell and are ultimately released. So the power of doing a CRISPR screen is that you can probe the host cell environment and try to uncover which of these host factors are actually essential for the virus, but not essential for the host. And this can potentially lead to some potential drug targets, and then also the ability to just discover new biology that was unknown before. So there are a lot of different kinds of CRISPR screens that are available these days. So I just want to explain the kind that we did and how we went about it. So we just took a CRISPR knockout approach. And to do this, we start with a population of cells, well, a cell line essentially that has Cas9 nuclease expressed. And then we take a library of single guide RNAs that are delivered by lentiviruses and transduce these cells at a low multiplicity of infection, such that each cell should only get one guide RNA. We use the full genome library so that every gene in the genome, about 20,000 genes, are targeted by 4 different unique guides. And then over the course of about 10 to 14 days, we select and expand these cells. And what we're left with is a very heterogeneous population of cells where every cell should contain a different gene knockout. We then apply the challenge. In this case, it's a coronavirus infection. And coronaviruses in culture are cytopathic. And most of the cells upon challenge are killed. So there's very few cells left. But those cells that are left, eventually they survive and they grow up to form colonies. And the expectation is that these cells survive because in part they either could not be infected in the first place, or they could get infected, but the virus could not replicate well enough to kill the cells. So we can isolate these cells, and then we can take the sgRNAs that we delivered by lentivirus and sequence them, and then use bioinformatics to go back and understand which are the host factors that the virus required. Here's an abbreviated depiction of the coronaviridae, just showing some well-known coronaviruses that infect humans. And obviously we were interested in screening SARS-CoV-2, but there are a number of other human coronaviruses that circulate. And we also decided to include not just SARS-CoV-2, but to include additional members from the alpha and beta coronavirus family, with the hope that host factors that we identify that are commonly required by multiple coronaviruses may not only be interesting, and useful for this pandemic, but also to give us more preparation for potential spillover events to identify host factors that might be more broadly required for the family. So I'm just going to go over very briefly the screens in general. There's a lot of data here. And also in the last symposium, our collaborator, JT Poirier, who we did all these screens with in collaboration, probably did a much better job than I would of presenting the screens. So, but I'll give you an overview of this before we dive into our favorite protein, which is TMEM41B. So shown here on the left are the 2 screens that we did for alpha coronaviruses with 229E on the top and NL63 on the bottom. And what I'm plotting here is all across the x-axis is an arbitrary gene list, arbitrary ordering of all the genes in the genome, where each dot represents a different gene. And then on the y-axis is the z-score is plotted, so that anything above or below these dotted red lines is considered significant. And there's additional information here in the size of the dot. So one is just, there's a number of hits, but one of the things that's always nice to see is that if a receptor is known, in this case 229E, the known receptor is ANPEP. So it's always nice to see that's coming out as one of your top hits. That's a good indication that your screen actually worked. And then also we can see down here that ACE2 is a very strong hit for NL63, and that is also the known receptor for NL63. Over here on the right are the results from the beta coronavirus screens with SARS-CoV-2 listed on the top and then OC43 on the bottom. We've added some additional coloring here to highlight some pathways that are prominent that popped out. In the orange color, you can see that there's a number of genes that are important in the SRABP control of lipid biosynthesis that were emerging as hits, as well as in the dark blue, genes involved in heparin sulfate biosynthesis. Heparin sulfate is known to be important as a non-specific attachment factor for viruses, so this comes up in a lot of viral screens. And then also genes which are involved in cholesterol biosynthesis. So like I said, you can look for your favorite gene and do a lot of Googling, and we did a lot of that. But another way to analyze the data is to take more of a pathway analysis approach. So just one example of that is what I mentioned in the previous slide is this pathway SRABP control of lipid biosynthesis. So this pathway includes a number of genes which are involved in the transport in and out of the ER of cholesterol. So it regulates cholesterol homeostasis. And shown here in heat map form are the results from the different screens that we did. Each column is a replicate, so each screen was done in 3 replicates. And you can also see that SARS-CoV-2 is listed twice, and that's because we screened SARS-CoV-2 at 2 temperatures, at 37 and 33, whereas the other viruses were screened at 33. And you can see that there's significant enrichment in this pathway, but predominantly in the beta coronaviruses, although there is some enrichment in the alpha coronaviruses. So we wondered, the alpha coronavirus screens were a bit more stringent. And one of the caveats to doing a full genome screen is that it's technically challenging to probe 20,000 different genes at once, although it does give you a less biased approach. But another way to perform CRISPR screens is to take a more focused approach, and you get additional insights that way. So we also collaborated with Francisco Sanchez Rivera from Scott Lowe's lab at Memorial Sloan Kettering, and Yadira Soto Feliciano from Dave Alice's lab at Rockefeller. And they, very early on in the pandemic, they took a paper that was published from the Krogan group, where they had done a protein-protein interactome study. And Francisco and Yadira had designed guide RNAs and cloned a specific library to probe in great depth every protein which was predicted to interact with SARS-CoV-2 proteins. And you can see from that result that now we see certain members of this pathway, particularly SCAP, are also showing up as important for alpha coronaviruses. So we think there's a very high likelihood that this pathway is very important for coronaviruses in general. And then at the other end of the spectrum, you see things like TMEM41B, where it's a prominent hit in all of the coronaviruses that we screened. However, if you try to do a pathway analysis on this, TMEM41B is involved in the autophagy pathway. And when we look at all other members of the autophagy pathway here, we don't see very much enrichment in the pathway in general, although there are a few genes which are important for some viruses. So, but TMEM41B is important at the earliest stages, again, for lipid mobilization. And we think that this might give some hints as to the mechanism. And I'm just alluding a little bit to what Heinrich will talk about in the next part. But we wanted to validate these findings for TMEM41B and the coronaviruses. So to do that, we had wild type cells. We have TMEM41B clonal knockout cells. So these are completely lacking TMEM41B. And then we reconstituted these cells with a cDNA to restore TMEM41B function. The cells were infected, fixed, and then stained for a viral antigen. And shown here are the data from 2 different cell lines, HU87.5 on the left, and then A549 cells that were made to express ACE and ACE2 and TMPRSS to allow infection of SARS-CoV-2. And you can see that all cells all viruses infect the wild type cells. In the absence of TMEM41B, there's no infection. And then when you reconstitute TMEM41B, the infection is restored. So the next question is, what part of the life cycle might this be functioning? So for this, and also to get another view of what's happening, we did qRT-PCR after infection. And you can see that by measuring a number of different amplicons from the virus, that there's about a 4 log reduction of viral RNA in the knockout cells, which is restored upon TMEM41B addition. So we think that this suggests that the block is either occurring at entry or somewhere up to or at the step of virus RNA replication. So to ask the question of whether or not entry was important, we utilized a reporter virus that we have in the lab, which SARS-CoV-2, which expresses nano-luciferase, and we made RNA in vitro. And by making this RNA in vitro and electroporating it directly into cells, this allowed us to bypass the entry step. So in this case, we could bypass ACE2 receptor or anything involved in getting the virus in. And you can see from this data here that even if we completely bypass entry and we deliver the RNA directly to the cells, that the virus RNA replication is severely impaired in the knockout cells. So this is really only just the beginning of getting hints at what the mechanism might be. We know that TMEM41B is important for lipid remodeling, and we also know that it's not happening with the entry step. But there are some additional insights that we have from previous work that we're doing at the same time for flaviviruses, even though it's a very different family of viruses. So I'm going to turn it over now to Heinrich, who will give you some updates on that story.
Right. Good morning, everyone. So I'm going to talk about the flavivirus aspect of TMEM41B. And so a year ago, COVID hit us, and we started to work on coronaviruses. But before that, we had an emerging virus, namely Zika, a few years back, and we did also genome-wide CRISPR knockout screens, where we probed Zika virus and also yellow fever, because we are a flavivirus lab and we have a long-standing interest in yellow fever. And among all the hits, we identified TMEM41B, and there wasn't much known about this protein, and we started to work on it. So Bill already alluded to the autophagy pathway, and similar to the coronaviruses, we find for Zika and yellow fever, TMEM41B was highly enriched together with the BMP1, which is its partner in crime, but none of the other autophagy genes were hits. And it becomes even more interesting, or if you do a cross comparison between our Zika screen and the previously published autophagy CRISPR screen. So you have the hits here, classical autophagy genes, our hits, and the only overlap between those two screens are TMEM41B and BMP1, highlighting that maybe the autophagy pathway might not be as important for the virus, but rather TMEM41B and BMP1 for its function in mobilizing lipids. So first we went on to, of course, test and confirm whether TMEM41B and BMP1 are host factors as they were identified in the screen. And we made CRISPR knockout clones for TMEM41B and BMP1, and we also expanded it to TMEM41A and TMEM64. And we did so because these four genes share homology in a certain domain, which is called the VTT domain for BMP, the TT for the 41, for the TMEM41s, and then TMEM64. But what becomes obvious is that if TMEM41B and BMP1 is missing, viral replication is drastically impaired relative to wild type cells. So I went on to take one of the TMEM41B knockout clones, reconstituted with all the different VTT domain proteins, and just to show that reconstitution with TMEM41B is completely rescuing viral infection. But we're also seeing that overexpression of BMP1 in the absence of TMEM41B can partially rescue infection of Zika virus. And similarly, we took one of the VMP knockout clones, and overexpression only of VMP1 can rescue infection. So we confirmed that VMP1 and TMEM41B are host factors, but then we also wanted to go on to see how much of the autophagy pathway is actually needed. So we made CRISPR knockout clones for some key genes in the autophagy pathway, BACLAN1, ATG5, ATG7. So if these genes are missing, the autophagy pathway is basically not functional. But as you see here, knockout cells replicate the virus just fine relative to wild type cells. And again, it's highlighting that the autophagy pathway or functional autophagy pathway might not be as important, but rather TMEM41B and VMP1 and their function to mobilize lipids. So I'm showing here a phylogenetic tree of the flaviviruses, one of the genus from the Flaviviridae, and you can divide it into mosquito-borne flaviviruses and tick-borne flaviviruses. And aside from Zika virus, we also tested yellow fever, dengue, and West Nile, just to see how broad the impact of TMEM41B for flaviviruses is. And relative to wild-type cells, you see that in knockout clones made with 2 different guide RNAs, the virus is highly impaired in knockout cells relative to wild-type cells. And in collaboration with the CDC, we were also able to test some tick-borne viruses. We had a group of viruses causing encephalitic disease and also hemorrhagic fever. And again, knockout cells don't support viral infection relative to wild-type cells. So this is all done in mammalian cells, but we were wondering how much is TMEM needed in the other host, like in the arthropod-borne host in mosquitoes, for example. So we made knockout cells in a mosquito cell line called AG2. And you also see here that if TMEM41B, the homolog of TMEM41B in mosquitoes is missing, the virus is highly impaired relative to wild-type cells. If we bring back the protein, we can rescue infection. Yeah, so this was for the flaviviruses, but the Flaviviridae also include other genera like the Heparin, Pegivirus, and Pestiviruses. And so we tested for the Heparin hepatitis C. The virus is impaired in knockout cells. And similarly, in Pestiviruses, where we tested BVDV, the virus is impaired in replication in these knockout cells. So Bill talked about the coronaviruses, and I just talked about all the flavivirus we tested. And we went on to see how important is TMEM41B for other viruses, and it's really not. So we tested a large family of different viruses, positive and negative sense RNA viruses, and there was no negative effect of these viruses replicating in knockout cells. Similar for DNA viruses or retroviruses, infecting knockout cells did not impact them at all. Also highlighting that the lack of TMEM41B is not really decreasing the fitness of these cells. So a fun fact, interesting fact, TMEM41B encodes for a SNP. And so position 266 is an isoleucine, but it can be a leucine or a valin. And it's prevalent in the Asian population, specifically in the East Asian population, even up to 20%. But this SNP is basically absent in the Europeans and African population. And if you take our knockout cells and reconstitute with a wild-type protein or with the 2 different SNP proteins encoding a leucine or valin in the position 266, we see that these cells support infection, but infection is rather diminished relative to the wild-type protein. And if you take then the soups of these viruses, of these infections, and titrate it, we can also see that the output is diminished in these SNP encoding cells relative to the wild-type cells. So I think I convinced you that TMEM41B is quite important for flaviviruses, but the question is, what is it actually doing? So similar to the coronaviruses, we took a yellow fever replicon, which is encoding firefly luciferase, and it's basically just RNA, which we transfect into cells. And then the filled circles, we have a wild-type replicon. In the open circles, we have polymerase dead replicon, which is our control. And if you transfect RNA for this replicon, you have initial translation of the incoming RNA. So you have an increase in firefly luciferase values. Then there's this RNA is then slowly being degraded, and there's a stealth period. And then around 12 to 16 hours, there's this huge signal coming up because the virus is replicating quite strong. And therefore, we have this increase in firefly luciferase for the wild-type replicon in wild-type cells. If you look for the open circles, the wild-type, the dead replicon in wild-type cells, the RNA is just being degraded over time, so there's no signal coming up. Then for the wild-type replicon in the knockout cells, again, you have this initial translation. So translation of the virus does not appear to be impaired, but then there's nothing happened for a long time, and then replication is highly impaired relative to the wild-type cell. We can also see this if we infect actually with the virus. So if we infect cells at the HMOI and we measure viral RNA inside the cell, you can see actually quite a 100-fold decrease in viral RNA in knockout cells relative to wild type cells. But interestingly, if we measure the induction of innate immunity, you can see that these knockout cells, despite having very little RNA replication, there's this high induction of innate immunity indicated here by measuring OAS1, which is one of the key genes being upregulated as an interferon stimulated gene. And this is truly due to viral replication, because if we use UV inactivated virus, there's no upregulation of the innate immunity. So you need incoming virus in the knockout cells, which probably starts to replicate. Then this replication is somewhat abrogated or aborted, and this causes an upregulation of the innate immunity. And we also tested, or we wanted to show, that these knockout cells are not just super sensitive or alert to any stimulus. So wild type cells and knockout cells respond equally well to a known stimulus like the influenza virus Delta NS1 virus. So lastly, we inhibited the innate immunity. So we treated the cells with TPK1 JAK inhibitors, and while in knockout cell and wild type cells, we can boost replication and viral output quite strong. There's no rescue of replication in the knockout cells. So this indicates that the block of replication of the virus is due to the lack of TMEM41B, but not due to the upregulation of the innate immunity. So the innate immunity is rather a consequence of the aborted replication. So bringing this all together, that's our working model for TMEM41B for the flavivirus life cycle. And we think it's very similar to the coronavirus life cycle, as they are both positive RNA viruses, and they like to remodel membranes to build basically shielded replication complexes. So an incoming flavivirus, the RNA is being translated on ribosomes. The polyprotein is being inserted in the ER membrane, and we know that NS4A and NS4B of the flavivirus interacts directly with TMEM41B. And TMEM41B is also interacting with VMP1, and VMP1 is basically providing lipids to TMEM41B, which TMEM41B is then using to remodel membranes and to basically, in terms for the autophagy, to enlarge these autophagosomes. So we think in wild type cells, TMEM41B is hijacked, used by NS4A and NS4B, and these two proteins are known to cause membrane curvature and basically building this replication complex, which is, as I said, a shielded environment for the virus to replicate and to be protected from the innate immunity. And in TMEM knockout cells, TMEM41B is missing. The incoming virus is still being translated, and replication is probably starting, but then it's being aborted because it can't build this replication complex. So the RNA is being readily detected, and therefore we have this upregulation of the innate immunity. So with this, I'm going to end this presentation. I just want to acknowledge the people who are all involved. For the TMEM project, this is the pre-COVID virology group, Charlie, our mentor, then all the people who were involved in the TMEM project. Bill and I were working together on this, of course. Highlighting Kate, she was responsible for making the mosquito knockout cells, and she just published a paper on this if people are interested in this. Then also our collaborators, JT and Lindy from MSKCC, and Laura McMullen at the CDC. Then the COVID-19 group, during the shutdown, we did all the coronavirus screens in collaborating with JT from NYU, then Yadira and Francisco, as Bill mentioned, from MSKCC and Rockefeller. Volker Thiel provided us with advice and materials, and he's in Bern in Switzerland. Then highlighting Joe, he did all the graphing for the results from the coronavirus CRISPR screens, and also Alison, who got the BL3 up and running so that we can actually work in the BL3 and did all the work. And then finally, our funding sources. Thank you.
Thank you, Bill and Heinrich. This is great presentation. So I may have missed this, but have you checked whether the TMEM41B protein actually from the recent data on mass spec with the pull down with the viral proteins that the Nevin Krogan's lab has put together, whether there are any specific interaction with viral proteins.
So we haven't seen a pull down of TMEM41B for the coronaviruses. There was a recent paper 2 years ago for flaviviruses, and specifically for Zika, and they pulled down NS4B, or with NS4B they pulled down, for example, TMEM41B. But for the coronaviruses, we haven't seen any direct interaction with the viral protein. But we would assume that since it's probably remodeling membranes, and the proteins in the coronavirus, like NSP4 and 6, I believe, which are important for membrane remodeling, that they might interact with TMEM41B, but it has to be shown.